Welcome to this explainer, where we're diving into the incredibly high-stakes world of sterile pharmaceutical manufacturing. If you've ever had an IV fluid or received an injectable medicine at a hospital, you were relying on an invisible shield of sterility. And today, we are going to look at the rigorous, frankly unforgiving, tests that ensure that shield is absolutely flawless and that patient safety is guaranteed. So, it's a fascinating question, right? How do we actually prove it? Well, before a single drop of a life-saving injectable ever makes its way to a hospital pharmacy, we have to relentlessly stress test the exact environment where it was made. We absolutely cannot just assume a clean room is working perfectly. We have to prove it through a highly controlled, simulated trial. Okay, here is our roadmap for today. We'll be covering 1. The aseptic stress test, 2. Designing worst-case scenarios, 3. Executing the media fill, 4. Monitoring the sterile environment, 5. Forensics of a failure, and finally, 6. Maintaining the sterile shield. Moving right into Section 1, the aseptic stress test, simulating high-stakes manufacturing. Let's define exactly how we pull off this simulation without risking actual, highly valuable medicine. In the industry, this is called a media fill, or an aseptic process simulation. I like to think of this as the ultimate crash test. Basically, instead of filling vials with a life-saving drug, we completely swap out the drug for a highly specific microbiological growth medium. We are literally intentionally introducing a substance designed to encourage bacterial growth into a room that is engineered to completely eliminate it. It is the ultimate test of the entire facility, the equipment, and the personnel. You can see that nutrient-rich broth right here. It's Soybean-Casein Digest Medium, usually just called TSB or Tryptic Soy Broth. We substitute this sterile broth for the real drug, and we run it through the exact same tubing, the same filters, and the same filling nozzles as the real product. We just want to see if any contamination manages to sneak in under pressure. Which brings us to Section 2, designing worst-case scenarios, pushing the system to its limits. To truly test this sterile shield, we can't just simulate a smooth, perfect day in the clean room. Honestly, that wouldn't tell us much about where our controls might break down. We have to simulate the absolute worst conditions. We stack up a standard shift against worst-case scenarios. We're talking maximum workloads. We want the absolute longest run durations to really test operator fatigue. We intentionally throw in shift changes, power fluctuations, and really complex equipment stoppages. We even use the most difficult container sizes. Because if our process controls and our operator skills can hold up under that level of extreme stress, well, it proves the process is genuinely robust. All right, Section 3. Executing the media fill, the 4 critical phases. Let's look at how this procedure unfolds from start to finish. A successful media fill is systematically broken down into these 4 phases, and every single aseptic manipulation is mimicked. We go from the initial garbing and cleaning in the setup phase to transferring the media aseptically right down to the final evaluation. So once that media is transferred perfectly, we hit step 3, which is crucial, a 14-day incubation period. But we don't just, toss it in a warm room and forget about it. The vials are actually incubated at 2 entirely different temperatures. Typically, it's 20 to 25 degrees Celsius for the first 7 days, and then bumped up to 30 to 35 degrees for the next 7. This dual temperature approach is essentially a trap. It's designed to catch both fungal and bacterial microscopic survivors that might be lurking in the vials. And finally, the evaluation. We check every single unit for turbidity, which is basically a cloudy appearance that screams, hey, we've got microbial growth. And the acceptance criteria for this, they are incredibly strict, as they absolutely should be. Look at this. For smaller batches under 5,000 units, finding even a single cloudy, contaminated vial means an automatic overall failure. 0 margin for error. 0 contamination is the only acceptable result. For the larger batches, just finding 1 unit immediately triggers a massive investigation. Section 4, monitoring the sterile environment, real-time safety nets. Because while the simulation is running, there's actually a whole secondary layer of testing happening at the exact same time. We have this invisible safety net actively monitoring the clean room itself. We use a really strategic mix here. Active air monitors pull volumes of air right through slit-to-auger devices, while passive settle plates just rest quietly on surfaces. We use surface swabs all over the equipment. And, maybe most importantly, we do gloved fingertip and thumb testing on the actual human operators. We're actively hunting for both viable microbes and non-viable particles floating around the room at the precise moment the simulation is happening. To put that into perspective, look at these razor-thin thresholds. Finding more than 3 colony-forming units, or CFUs, on an operator's gloves after a media fill or on a critical work surface triggers an immediate action level. Just 3 tiny colonies. That constitutes an overall failure of the operator's aseptic competency. Okay, Section 5, Forensics of a Failure, Investigating Microbial Growth. So what actually happens when the crash test fails, and we spot that dreaded cloudiness in a vial? We immediately pivot into a systematic, full-blown forensic investigation. Step 1, immediate quarantine of the batch. Production halts entirely. Then we backtrack. We comb through operator gowning logs. We scrutinize all that environmental monitoring data we just talked about, and we rigorously verify the HVAC airflow and equipment calibration. We'll bring in root cause analysis tools, like the 5 whys or fishbone diagrams, to pinpoint the exact source of the breach. Was it just a microscopic tear in a glove? A sudden drop in air pressure? We literally cannot move forward until we find out. And that leads to Section 6, Maintaining the Sterile Shield, Preventative Action. Because finding the root cause, that's really only half the battle. Uncovering that cause leads directly to implementing HEPA, corrective and preventative actions to fortify the process so it doesn't happen again. You see, passing a media fill once isn't the finish line. Maintaining that state of control is. Preventative maintenance is our absolute frontline defense. We're talking daily visual checks and routine physical integrity tests for isolator gloves. Because a faulty glove is a direct highway for contamination. It means constantly revalidating massive HEPA filters, checking cleanroom airflow, and, crucially, it requires rigorous, never-ending personnel retraining. So, why does all this matter to us? Well, these strict ISO classifications, these exhausting forensic investigations, and the rigorous worst-case simulations, they're the exact reason you can trust the sterile medicine you receive. This whole process is what ensures that the invisible shield protecting the product, and ultimately protecting you, remains unbroken. I want to leave you with one final, kind of provocative thought. Throughout our sources, one variable just continually pops up as the absolute greatest contamination risk: the human operator. So, as automation, closed isolators, and robotics continue advancing so rapidly in pharmaceutical manufacturing, is the human element the next thing to be eliminated from the clean room entirely? It's a truly fascinating frontier for the future of medicine. Thank you so much for joining me on this explainer, and keep questioning how the world around you works.